One of the things with the early precursor disease and smoldering that I wanted to highlight was this concept of moving of the risk scoring and risk profiling of smoldering. So right now, all the risk profiling basically of MGUS and smoldering is based on the burden of disease. What is the M spike? What is the free light chain ratio? How many plasma cells are there? So it's basically just looking at how many, what are the labs and how many cells are there? So there are there's coming data now coming out on looking more about the biology of this and having a more biological base for doing these risk scoring. So Nicola Bolli summarized this in a very, very nice fashion where they have that whole genome sequencing for pairs of smoldering, those that have progressed and those that have not progressed. And those that have progressed, they do have a different type of genomic kind of landscape. So they have a much more complex genomic landscape. They have more hotspot mutations, they have more deletions, they have more complex structural variations, so the chromotripsis and chromoplexes, and also they have the difference in the mutational signatures, which are much higher profile of the apobec signature. So this is basically, this is right now on the research side, but this would be very important to be able to put this into a clinical test and do this type of clinical profiling, because this, then we can risk profile patients much more on the biology. So instead of moving from MGUS to smoldering to myeloma, we can have kind of non-progressors and high risk for progression. And those patients in the next step would be those that we could have early interventions, early trials. So I think that was some of the very interesting updates that came out of that.